One or AS Biology video. This week we're going to be looking at digestion and carbohydrates. So first of all, we need to know what digestion is. Well, digestion obviously uses lots of organs, so the digestive system is made up of lots of individual organs and they're highly coordinated to affect the whole body. Digestive system works by using enzymes to do hydrolysis reactions, which, if you remember, that means adding water, and it breaks large insoluble molecules into small soluble molecules that can then enter the bloodstream. And you need to be able to identify the names of the different parts of the digestive system. So the food goes obviously via the esophagus into the stomach, around the small intestines, through the large intestines, and then stored in the rectum for at least via the anus. Important parts in there as well. So what do we need to know? Well, this is just a bit of a brief overview. The mouth obviously is where mastication takes place, and that is where you chew your food and break it into smaller pieces and you mix it with the enzyme. Esophagus is obviously where food travels down, and it's where your muscular wall does this thing called peristalsis, where you swallow the first part of peristalsis, a muscular contraction that forces your food. The stomach is made up of thick layers of mucus releasing cells. It produces hydrochloric acid, and obviously it contains protease, which digests. And your small intestines is a long tube where all the absorption happens, but also enzymes involved in digestion of lipase occurs, and we're going to talk about sugars again today. So sorry, lipase is the enzyme that digests lipids. Pancreas, your liver, and the gallbladder. The liver and gallbladder produce bile salts that release and emulsify the fat, and the pancreas produces all the enzymes we're going to talk about, so lipase, trypsin, which is type of protease, and amylase. And the liver stores excess glucose as glycogen, and is also the site of deamination with excess amino acids. The liver is an essential job. Large intestines, or the colon, is where the water is reabsorbed or absorbed, into your bloodstream, and that dries out. So obviously, what we call, and then at the end, feces or the rectum until they can remove later on. That's called digestion, not excretion, because you don't actually. It's not a waste product. It's just undigested food. So then we're going to look at digestion of carbohydrates today. So obviously glucose is vital. It is the chief respiratory substrate in our body. So it's one of the things that's broken down for respiration, or oxidized for respiration. And obviously glucose comes mainly from carbohydrates. Glucose is small, so it can be absorbed immediately through the gut cell wall. But obviously starch itself, which is how we get most of our glucose in plants, starch is a long polysaccharide and it's insoluble, so it must be digested into maltose and then glucose using two different groups of enzymes. So basically there's two hydrolysis reactions that occur. So we're adding water to break down one larger molecule into a smaller molecule, and the first one is using the enzyme amylase, which is in our saliva glands, release saliva which contains amylase, salivary amylase, and that breaks down starch long chain insoluble polysaccharide into a smaller disaccharide called maltose, and then the enzyme maltase breaks down, and there's the word for maltase. The enzyme maltase breaks down maltose by again adding water, hydrolysis reaction, into two alpha glucose molecules, which then get absorbed into the bloodstream. So let's look at that in a bit more detail. So as we just said, the same thing there, okay, but now let's just have a look at so where these things happen. So in the mouth is where amylase digests starch into maltose. That amylase then will get denatured as it enters the high pH, or low pH, the high acidity of the stomach, pH 2, due to hydrochloric acid. And then in the small intestines, amylase again is secreted from the pancreas via a duct, via the pancreatic duct, into the small intestines to finish off any undigested starch and digest it into maltose. Now the maltose will be too big to go across the villi of the small intestines, so we have membrane-bound disaccharidases. So think about what that term means. Membrane-bound means it's stuck to the villi wall or the intestine wall, and disaccharidase means it breaks down disaccharides such as maltose. And the epithelial cell lining of the ileum, the first part of the small intestines, have enzymes that are stuck into the wall, which will digest those disaccharides, the maltose in this example, into glucose. Okay, and maltase is there ready to do that for us as the example. Obviously, to help absorption, once that is digested, we've got a large surface area because we've got villi, which are covered in microvilli. So here we've got the image of the villi in the small intestines, and if you were to magnify one of these, you would see the microvilli onto the small intestines villi, and that increases the surface area to maximize the diffusion and also active uptake of glucose in the intestine. And there you've got a picture of the membrane-bound disaccharidase. So the maltose comes in as it passes, it digests in the via hydrolysis reaction and turns into glucose. The disaccharidases have to be membrane-bound, otherwise they would obviously get passed down the intestines and passed out of the digestive system. And that. So what do we need to know? Well, you need to answer this question. So how is glucose digested from starch and then absorbed into the bloodstream, which could effectively be up to like a 12-mark question in the exam. And it really has three parts. So the first part is where does the digestion happen in the mouth, then digestion in the ileum, and then finally the absorption across the villi, which is an active process. Let's start off then. So if we start off with digestion in the mouth, we'd say amylase is released from the salivary glands and the pancreas. That is then causes a hydrolysis reaction, which is the addition of a water molecule that breaks down starch into maltose. Okay, and actually I'd add an extra thing in there. It breaks down glycosidic bonds. We then have the digestion in the ileum, which is where obviously you could say again amylase digests starch again, but membrane-bounded organelles, called membrane-bound disaccharidases, digest disaccharides into monosaccharides, and they're membrane-bound, so they're not pushed down the digestive system. And the example that we need to know is maltase digests maltose into 2 alpha glucose molecules via a hydrolysis reaction, which again breaks that glycosidic bond. Now, in terms of absorption, this is where we need to go down the route. It's very similar to protein absorption or amino acid absorption in the intestine. So if you look at this diagram, we've got the lumen of the small intestine there. So that is the small intestinal ileum, and then we've got the blood. And we want to get the glucose, which is the red molecules, from the small intestine into the bloodstream. But it's not a straightforward process. So if we start off with the glucose molecules need to somehow enter there. So let's have a look how we do it. Well, actually, the first thing we deal with is sodium ions. And sodium ions, as with digestion of amino acids, sodium ions, or Na+, are found naturally in the epithelial cells. And what happens is the first thing is they are actively pumped out of the epithelial cells down into the bloodstream. What that does is it gives us a low concentration of sodium then in the epithelial cells that line the small intestine. And because we've got lots of sodium naturally in our food, because sodium, if you get sodium chloride is the proper name for salt, we've got higher concentration of sodium now in the small intestines, low concentration of sodium in the epithelial cells. So then the sodium wants to diffuse into the cell, but it can only diffuse into the cell if it moves through a co-transporter protein. And as it comes through this co-transporter protein, it also brings along with it glucose molecules. Okay, so every sodium that comes in, it brings glucose. That means then you end up with a high level, I'm just going to write G for glucose, even though we know it's C6H12O6. You then end up with a high glucose concentration in the epithelial cell, and then the glucose diffuses from a high concentration to a low concentration down through the carrier proteins and into the bloodstream. Okay, so again, just go through it one more time. Sodium ions in the epithelial cell are actively pumped into blood. This lowers the concentration of sodium in the epithelial cell, causing a concentration gradient because there's now more sodium ions in the ileum, and they diffuse across into the epithelial cells. The sodium then does facilitated diffusion by a co-transporter protein, bringing in the glucose into the epithelial cell, and then the glucose diffuses down the concentration gradient through the carrier protein or char protein, via facilitated diffusion. So you need to know that. You need to be able to apply that. Very similar to the absorption of amino acids. There are slightly different versions out there, so you might see slightly different versions, but as long as you use the picture you're given in an exam, you're always likely to be given something like that to support you with. Okay, guys, hope that's been useful. Thank you very much.